now we can get to the fun stuff. Yeah.
Where one sets off another.
What's that?
Where they all kind of set off each other,
Yeah,
like in
set
this
off
picture.
Each other. We're going to keep it real simple here.
Okay.
Which picture are you looking at?
This one. We kind of went over it yesterday in our review with Josh. It's kind of cool. I was like, oh, that's how all the signals get transferred. It's pretty fun.
Okay, so you have these channels in here. I'm going to walk you through what happens during an action potential. Okay? So, you have potassium that's going through the cell with a pump, right? So we have potassium being pumped in, sodium pumped out. And then maybe a ligand comes by and it attaches here. And it opens up the sodium channel. And so it causes the nerve to reach a charge of negative 50 millivolts, which is threshold, right?
Mm-hmm.
So we're going to see, let's see. Sodium is rushing in here. And the charge is at the right, it's at the right charge at the axon helic. And so if those channels there say, oh, it is negative 50, we can finally let in more sodium. These voltage-gated sodium channels will open. If they open, sodium rushes in. And you're going to see this whole thing occur. You guys recognize this from class? And then they switch. You guys recognize that?
Not from class necessarily, but I think I remember from the review of last week.
All it is that inside of the cell is becoming more positive, right? How it's coming up here.
And so is that like the axon becoming more positive? Is that what that picture is?
Yeah.
Okay,
so
that's
it's
what
a
I
nerve
figured.
cell.
Yeah.
And you see that. And so it's going to travel down like that along the cell. So these will eventually become more negative, while those become positive. And
So
those
eventually it
become
goes back to normal. It's just sending like a signal down.
Yep, it just goes. It doesn't make that sound, but it does do that. Does that make sense?
Mm-hmm.
So it just lets sodium in here, and it just kind of goes down all the way.
So the sodium's going all the way through the axon? Or
Yeah.
Oh, okay. Because
So
it's
you'll
going
have-
through the entire cell.
I don't know if they'll teach you. I think this is in the book. Well, actually, I don't know. It's definitely online though. But you'll have sodium channels here, which are voltage gated. So as you send a signal here, it says, oh, we're at the right voltage. More rushes in. Oh, we're at the right voltage. More rushes in.
That's
And it
sweet.
travels through it. So that's why myelin helps with saltatory, what do they call it, saltatory reaction, I guess, where it's actually able to like skip parts of the cell because it's myelinated, because it doesn't have to worry about leak channels here. They're all blocked. So they go here real quick, they jump, more sodium goes in, jumps, more sodium goes in, rather than just one slow continuous thing. Okay?
I'm just writing these down.
Now we're going to magnify this. That's going to be, we're looking at it over here. Okay? Magnification. So this is the nerve here, and the muscle here. So right here, you're going to see a voltage-gated calcium channel. This is where calcium starts playing a really big role in all of this. What calcium does is it attaches to snare proteins. So these circles right here are vesicles. Vesicles are just kind of like pockets that hold stuff.
Wait, the circles with the X underneath them?
Yeah.
Okay.
So this is the vesicle,
Okay.
The snare proteins, they're attached here, and then they attach to the inner wall of the nerve.
Okay.
Right there. And then inside of these vesicles, we have neurotransmitters. And neurotransmitters are chemical signals. So when they're released, they attach to a receptor, and they say, hey, something's coming in, something's going to happen, type of deal. So what calcium does is it comes in this way, and it attaches to snare proteins. And you see how those snare proteins are kind of like an X? Calcium pulls it down and twists up the proteins, and so that the vesicles are really close to the wall. And this is where we see exocytosis, which you guys have probably heard about. So the snare proteins are still there, they're just tightly bound down here by calcium. That makes sense? And then the vesicles open up, and acetylcholine is now released.
Wait, okay, so go back a minute.
Okay.
So, hold on. Now I'm just trying to see what question I actually have. So, it goes down the chain, goes down the nerve cell. And then we have neurotransmitters. Are they just kind of already, like, naturally in the nerve cell? Like, how do they get there?
Yeah. So,
Are
it's
they just made in there?
acetylcholine. It's called acetylcholine.
Okay.
And part of this is recycled from here. So it's naturally occurs in there.
Okay.
Or it just goes there. So when it separates, I think it's the choline that's, one of these is already in the cell, and the other one kind of gets dissolved, but it'll eventually come back and make acetylcholine. So your body just naturally puts it there.
Okay.
That makes sense.
A little bit. And then, so we have, what just made it release, I guess, is more my question.
So calcium bound to these snare proteins here. Calcium comes in and it comes around here and it pulls this down so that it looks more like this. And then it keeps pulling it.
And it pulls it out of the cell.
So it's like this. And then
So
when it's that
pushing
close
the
to the wall,
protein.
it just opens up.
Then what happens to the other protein?
The snare protein,
Yeah, the snare protein.
it's still there. Probably along the side. So once these
Oh,
are
so-
released, that'll close back up. And the snare proteins are going to relax because the calcium's released, and then it's back to normal. And
Now,
this-
does the calcium go out with the vesicle then?
No, I think it's still attached to the snare proteins.
Okay.
It'll
then-
probably get released through like calcium leak channels.
Okay. Because I was wondering, like, just like if it were to just stay in there, then the cell's gonna get more positive with the buildup of calcium. So I didn't know how it.
Yeah, I think it just leaks out.
Okay.
So. Any questions, Neil,
Makes sense so far.
so far? Yeah.
Except for why is everything we talk about with acetylcholine?
Because we're talking muscles right now.
And that's just like the main factor, I guess, in everything they do.
Acetylcholine plays a big role with nicotinic receptors and muscarinic receptors,
Okay.
which you're going to learn about in a little bit. But I think it might be the next test. But acetylcholine we're just talking about because that's to do with muscles, and it's like
Okay.
a real common one.
There's another neurotransmitter that we talk about too, but it's like it's the N word. It starts with an N.
Nicotinic?
No.
Nicotine?
No. North.
Norepinephrine?
That's the
epinephrine?
one.
It's like
Norep-
adrenaline?
Yeah, something like that.
Yep.
There's one other. That's the other one that we talk about. 'Cause I know because it was in the reading and I wrote it down. And I was like, that's such a weird word.
It's pretty much like adrenaline, kind of.
Okay.
Okay.
When do we learn about that one more specifically?
I think it's the next test. I'd have to read up and see when it is. Super interesting stuff, though. That's when you start learning about medicine and how it plays a role. Like, when you watch a commercial and it's like, take this medication may cause drowsiness, death, swelling, it goes on and on and on. Because it blocks a certain receptor not allowing those things to take place, or it activates a receptor so that all those things take place. So like smoking has nicotine and it affects a lot of your receptors. So it increases your heart rate, causes vasoconstriction, causes your lungs to constrict, like it just goes through all these different things. That's why smoking's really bad for you. Plus it's smoke going in your lungs. So yeah, it's interesting. It's fun stuff. You guys will like it, I think. Okay, any questions with this so far?
Don't think so.
Okay, this is about the point where we're going to stop. I'll let your teacher teach the rest. So here you have a ligand-gated sodium channel.
And then it just starts the whole process over. Well, it's muscle now.
So for this is muscle, so I'm kind of teaching the end part.
Okay.
Okay, so these acetylcholine, it's a neurotransmitter, right? It's a chemical signal. It's a ligand. So these actually set off ligand-gated sodium channels. So you'll have some of these come here, and they'll sit down on the voltage or the ligand-gated sodium channel, and then it's going to activate it. So the sodium that's out here is now going to rush in, changing the voltage of the muscle. And right here-ish, we have a voltage-gated potassium channel. And so if the voltage here is changing, it's going to activate the potassium channel, and potassium is going to rush in, or if the action potential, sorry,
No,
action potential is
rushes out.
going to rush out. So we'll get into that in just a second.
Sorry, continue.
I might have
Hold
taught
on.
that in the wrong order.
So
Yeah.
are you saying that the potassium channel is like in between the muscle?
Yeah, so this is the same muscle. You guys learned about T2 wheels yet?
I don't think so.
Maybe I'm teaching too far. We'll call it good here. Yeah?
I'm so
We'll call
excited to learn though.
I know, I let your teacher teach that first. Okay,
Dang it, okay.
so action potential, I'll teach the real simplified version.
Okay.
Action potential travels down here. There's something called the SR, or the sarcoplasmic reticulum. It releases calcium, your muscle contracts.
Sweet. Yeah, we might
Deal?
be talking about that next round. when we
You'll talk about
muscle.
actin and myosin.
I think that's when we do start the muscle.
Yeah, that's actually I think Friday.
Is it?
Yeah,
Perfect.
I think I read
That
that.
is the nervous.
Come in on Friday. That's the fun stuff. You guys are learning the best stuff in class right now. Okay, does this make sense?
Yeah.
So what you'll actually have is a nerve. Oh, I'm drawing something else. Well, no, that actually works. Travel to a nerve, travel to a nerve, travel to a nerve, so on. And then it's going to hit your muscle and cause a contraction. That make sense?